Very good morning, students. So this is the first class of advanced clinical biochemistry, but I am going to handle this subject. It's a very important subject, okay, in biochemistry, MSc biochemistry major. And first of all, okay, I would like to show the syllabus to you. I will share my screen. Okay, you will get a pop-up window. I will share my screen, entire screen, and the screen will be appearing. Okay, maybe Vinod Kumar is presenting. You are presenting your screen like that. Okay, just you will get a window. In the center of the window, just you can pin it. You can see here this pin button is there. Can you able to see this window? Just you can type it in the chat box. If you're not getting, just type no. Yes, good. Can you able to see everyone? Yeah, this procedure you already followed, okay, in the webinar. The same thing, okay, this the same platform we are going to use it. Okay, this is the pin. Just you can pin the screen and you will get my slides and so all the PPT and everything. Okay, so this is your recent syllabus, that is the 15th board of studies. We passed advanced clinical biochemistry and molecular diagnostics. This is the heading of your subject, and the main objective is to bring all the components, okay, within this advanced clinical biochemistry. And here the first unit will cover all the basic parameters, that is the different phases of the clinical laboratory procedures. So this is the real subject. Actually, this is the core subject of biochemistry. If you are entering into the medical laboratory or otherwise the clinical laboratory, so what are the phases you are going to maybe you are going to face it, the phases to be faced. So there are three phases. One is the pre-analytical stage, analytical stage, and the post-analytical stage. So before the analysis, and during analysis, and after analysis. So all the three segments will be covered in this unit, so which includes everything. Okay, this will give you the clear picture about all the informations related to clinical laboratory procedures, the collection of specimen and processing of specimen, and during the analysis, okay, what are the steps to be done, okay, that is sample preparation and reagent preparation, mixing and everything, okay. And general principles and interpretation of laboratory results, okay, maybe after completion of your test, and what are the informations to be given to the students, like general principles and interpretation of lab results. So how we can adopt the methodology to follow the statistics. For example, if you are getting the result, if you are doing only one time, for example, if somebody is giving the blood sample to analyze what is the serum glucose level, if you are doing only one time, so what will happen? So that one time you are getting, okay, 80 milligram per deciliter. Whether that is true? Do you think, okay, that is true? No. Actually, you have to do it in triplicate. So 3 times you have to do it as per the statistical norms. And all the 3 times you are getting, okay, 80 or 81 or okay, 78, like this. If you are getting, okay, the values, you have to take the standard deviation or standard error mean. So finally, based on the 3 values, if you are getting 1, so that is called as the real value, the result. If you are getting the first result is 80 and second result is 40 and third result is 120, do you think, okay, this is right? What is the procedure, okay, you are adopting it, and the results you got in 3 experiments, okay, which is correct? No. So this something is fishy and something is wrong, okay, in your experimental protocol, and you have to reschedule it, and again you have to do it till getting the similar numbers. For example, I am not saying, okay, the 80 means 80 you have to get it. It is not practically possible at all. Okay, if it is 80 and another one is maybe 82 or 85, and maybe the lower range may be 75. So the plus or minus 5 milligram you can accept, and usually in the industry, that is a clinical lab industry, and the acceptable parameter is only 2 mg, that is plus or minus 2 mg. So that is a very stringent and also very strict rules, okay, they are following it. So these are all the informations to you, and you have to follow all these procedures. You are not permitted to do only one time of assay. And if you are loading the sample, if it is auto analyzer, if you are loading the sample automatically, that will do 3 times, at least minimum 2 times. Okay, you can ask the question, sir, 3 times maybe we can get the value of standard error mean or standard deviation. and maybe if you are using two values, for example, if you are doing 2 times due to saving, we are all Indians, no, okay, we will do a lot of homework, okay, to minimize the cost, okay. So you can use 2 methods, maybe the first method and second method, maybe the first test and second test. First test if you are getting 80, and second test if you are getting 82 or 79. If it is this values or this range, it's okay. The first one is 80 and the second one is 120. So which one is right? The first method and second test. So here this may be the problem. That means, so we cannot choose, okay, which method is right. Again you have to repeat it. So this is the drawback of getting maybe working with the 2 methods. As per the statistics, okay, we have to do 2 methods, sorry, 3 methods. That is test 1, test 2 and test 3. Test 1 is 80 and second one is maybe 40 you are getting, and third one is 82. So the first one, last one is right, and you have to remove the value of the middle one, and you can do the standard error and mean or otherwise standard deviation, and you can give the report. So this is the one, okay, you are going to face it really in the industry, that is the clinical lab industry, okay. So now you have a lot of scope, okay, due to COVID-19. I hope, okay, everyone is fine and your family members are fine. At the same time, okay, you have a lot of scope, okay, due to this COVID-19, because everybody realize, okay, the importance of biological sciences, because you are the one, okay, going to deal with all the samples, okay, in the clinical lab, and if you know the techniques of qPCR and also the processing of samples and everything, you will get a job surely, okay, in the industry. Some of your seniors, okay, they already got the job. You know that, okay, some of your immediate seniors also, before getting the degree, they got the job, okay. So this advantage of the one side, okay, we have the lot of troubles, okay, with COVID-19. At the same time, another one is it gave some knowledge or otherwise some awareness to all the public. So some of the subjects are like very important, okay, like biochemistry, microbiology or biotechnology. So you have a lot of scope, okay, in the near future, and so this is covering all the aspects, the introduction to clinical laboratory procedures. So this is not going to be handled by me and the another member, okay, you know. So for you, I'll say that, okay, this is the workload. Second PG BC, this paper will be shared with KVA. Anupama madam, okay, going to take, okay, this paper along with me. Okay, right. And second unit is I'm going to handle, okay, this second unit. So this is very simple, the clinical chemistry based assessment of human nutrition and digestive function. So in the maybe the clinical biochemistry can be studied in different aspect. One of the aspect is this one, and this is the one, okay, internationally accepted. Okay, I was in Libya, okay, I taught same paper to the medical doctors and also MD biochemistry students. So this one is very important, because this concept of studying clinical biochemistry or clinical chemistry, organ wise or otherwise, the organ function wise, maybe, okay, you can study the digestive function, the liver function, okay, everything is the same component, okay, that means the digestive system. Any abnormality in the digestive system, okay, can be studied, okay, by using this technique, okay, and this is covering, okay, all the human nutrition and the digestive function and also liver function. And the third unit is the clinical chemistry based assessment of cardiovascular system. Cardiovascular means you know, the very well know that, okay, the function of heart, circulatory system, okay, and renal function, so that is excretory system, okay, you already studied in human physiology. So this is only the application part. And the fourth unit is clinical chemistry based assessment of water, electrolyte and acid base balance. So this one is the core component of biochemistry, and you are going to study about what is pH and how pH is maintained, okay, in the body, and what are the electrolytes like sodium, potassium, magnesium, chloride and everything. All are very important for the body's function. And next one is the mineral and bone metabolism, like the metabolism of calcium and how it is deposited in the bones, and so what is the function of bone, and maybe the how the hormones play a vital role, okay, in the calcium deposition and also, okay, the calcium dissipation, okay, both the functions, okay, you are going to study. And the last unit is clinical chemistry based assessment of human cancer. Cancer is a separate topic and separate unit, okay, given to you as a biochemistry student. So you have to study all the different types of cancers And so here in this paper we are going to study how to diagnose the cancer. Okay, we are not going to study what is cancer and what is the function of maybe what is abnormality in the cancer. Okay, not like that. So we are going to study everything, okay, like this. And the clinical significance of human cancers, all the markers you are going to study, the carbohydrate markers and blood group markers and everything. And these are all the textbooks. The main textbook is Teed's textbook of clinical chemistry. This is a main book. This is a Bible of clinical chemistry. And another one is Henry's clinical diagnosis and management by laboratory methods. And the reference books are there. The textbook of clinical chemistry and molecular diagnosis, and clinical chemistry techniques, principles and correlations, and clinical chemistry by Marshall, and the manual of laboratory and diagnostic test. So all these are the textbooks and the reference books. So most of the books are available in our college, and some of the books, okay, if it is not available, okay, just inform me, okay, I'll give the content. So we have the soft copy, and from the soft copy we can extract the pages and give it to you. And usually, okay, I will give my notes to you, because I wrote a book, clinical chemistry for medical and medical lab technology students. So that is the textbook, okay, for the medical college students at Libya. The same content, okay, I'm going to use it, okay, for your class as a study material. You can study from that book. That is more than enough. But don't be very specific to one book. As a biochemistry students, you have to refer 2 or 3 books. You can take very important points, and you can prepare the syllabus and study. Okay, and that is very important. Okay, if you are studying only one book will not cover all of your syllabus. So this syllabus is mainly framed, okay, based on the Henry's book. So this Henry's clinical diagnosis and management. I'll show the book maybe after this class during the discussion, and that's all about the syllabus you are going to study. And so this is very important paper, and frankly speaking very interesting also. At the same time, okay, you have to study each and every day. If you are accumulating everything before model exam or internal exam, opening the book and studying it, you cannot understand anything. So my main objective is recording this class, and I will post in the YouTube, and the link will be shared to you. Maybe if you're not confident enough to face the examination, just you can recollect all the information taught in the classroom. Okay, that is the main objective. So that is the advantage of handling the online classes, and the classes mostly with the laptops and e-content. Okay, so this is the advantage. Clear? I hope, okay, everyone is listening. Okay, and we can go directly into the content. Okay, this first unit will be handled by Anupama madam, and we can go to this topic. So today is a very auspicious day, and so we can start our classes. First one is the clinical chemistry based assessment of human nutrition and digestive system. So this human nutrition, how the human nutrition is functioning, that is the digestive system. You already studied human physiology in your UG level, so that is a base, and have some knowledge about pathology, human pathology, because both the information, the knowledge about the human system, okay, the human physiology, and second one is pathology. So these two are the base fundamental, and you are going to study the top, okay, that is the diagnosis part. So we are going to focus only the diagnosis. This is not my part to teach the pathology aspects or basics. So what is stomach, and how the digestion takes place, and what is the enzymes of salivary gland secretion, and what is the function of pancreas. This is not a job of teaching human physiology or pathology here. You already studied as a UG biochem students, and now you are in the second year, and it is your duty to have some basic knowledge. So we are going directly into the laboratory test, and maybe I will say some very important points about the basics. That's all. So first we can focus on the starting material is digestive system. So in digestive function, we can select the first organ is stomach. So you know that, okay, stomach is a very important organ, because that will get the food so directly from the mouth, okay, via esophagus. Esophagus is a tube, so that is connecting your mouth and also the stomach. So that is a very important organ, because esophagus, that is very sensitive also, and that will do some peristaltic movement and pushes the food into the stomach. You know, the main the type of cancer that may kill a patient very fast manner, and also till now, okay, there is no diagnostic test, and also there is no treatment is available, okay, for the particular type of cancer. That cancer is esophagus cancer, because esophagus is very sensitive. I said is a small, very sensitive tissue. That tissue will do the peristaltic movement and passes all the food particles from your mouth. After chewing, okay, you will pass your food, okay, into the stomach. So this process is happening, okay, in the real time, and automatically after reaching the stomach, automatically, okay, this stomach is having a valve, okay, that is attachment of esophagus and stomach. There is a joint is there, so there some valve is there that will close it. So that is why you will not get the food from the stomach to the mouth. Okay, the reverse peristaltic movement is not going to happen. Okay, somebody, for example, if you are not taking the food on right time, and also you have a lot of stress, due to that you have some acidity, and also due to that this over acidity or the hyperacid secretion may damage the valve. So this may liberate the loosening of that is valve, and also liberate more acids into the esophagus. You know, esophagus doesn't have the capacity to withstand acid and all. Only the stomach we have the protective layer. That layer withstand acid secretion. Maybe you can have the acid, okay, maybe pH 3 also, okay, no problem. At the same time, the esophagus is getting a slight acid, okay, into the esophageal region. So that will be damaging the tissues, and also all the superficial epithelial layers are damaged. If the damage is maybe acute, means if it is acute, no problem. Maybe within a time span that may be again regenerated or otherwise repaired. If it is continuous, for example, if you are not taking any medicine for your ulcer, and if you have the reflux, maybe acid is coming to mouth, or otherwise if you feel the heartburn, so this will create lot of problems, okay, to the esophagus, and automatically due to the irritation, the cells of esophagus will be turned into cancer. So this type of cancer, there is no treatment, and we have to remove esophagus, and maybe your mouth can be connected with a tube or something, okay, to the, and you know, this will create lot of problem, or otherwise they will cut the esophagus, and they may give a hole, okay, in your stomach, and you have to pour the food, okay, the liquid food, by using a funnel or something. So this is the only remedy, and we have to remove this esophagus. So without esophagus, living the life is very, very difficult. So that is why the God gave the gift, the healthy life, and we have to live a healthy life, and we can follow the system, okay, given to us. Okay, the first organ is stomach. I said, okay, the stomach is very important organ, and the main 25 to 40% of the digestion is happening only in the stomach. If your acid secretion is normal, and maybe if you are taking the right food in the right time and with the right content, okay, that is also good. If you are taking the right content, means why you are specifying the content, means if you are taking the normal diet, the normal quantity, because you know that, okay, what is the physical activity you have, how many hours you are going to walk, and how many hours we are going to spare some physical exercise or walking, or maybe doing the job or something. If you are sitting, okay, maybe so during this period, okay, most of us are sitting, okay, maybe the faculty members in front of the system only. So at the time, okay, we have to restrict our diet also. If you are having the same type of diet, okay, maybe going to the college and teaching the students the same diet, if I am taking, okay, what will happen? I will have some other abnormality, because sitting idle, maybe sitting in only one place and doing the job is so that is you know, without any physical exercise. We don't have the physical exercise. Okay, in the time, okay, we have to take only 25 to 50% of the food. If you are taking like that, no problem. At the same time, if somebody is taking the over food, okay, over intake, means so the 75% of the stomach will be filled, and the acid is secreted more, and due to the more acid secretion, all the complications will come. Okay, that is ulceration of the tissue, and reflux. Reflux means if the stomach volume is normal, no problem, because it will not come out of the stomach. If it is over volume, automatically due to the compression process. So because the stomach is having a lot of muscles, and all the muscles will do a mixing job, and maybe after secretion of acid. Okay, if the food content is more, automatically due to the pressure, the positive pressure, and the food will slowly come out of the wall and reaches esophagus. So all these problem will come, and the main problem of stomach is acidity, and this acidity is not a maybe diagnosable one. Okay, the acidity is very common, okay, in the younger age, and especially the students, and also the persons going for IT job and all, because stressful jobs, okay, you will give a lot of ulcer, including I have ulcer because due to my stress. Okay, so we cannot avoid ulcer, but we can keep it as under control. If the ulcer is not controllable, automatically that will harm all the parts of the stomach, and this may lead to some other complication like cancer and all. So we have to be very careful, and the prolonged or the chronic ulcer, that may lead to cancer. So we have to do some endoscopy and check, okay, what is the pathology of the stomach, and if it is more ulceration, okay, we have to take some tablets like omeprazole or otherwise pantoprazole, okay, with anti-emetic substances like some of the substances are available in the market, and maybe the dual dose is also there. And this is the main problem, okay, to the stomach. And we are coming to the main part, okay, that is the infection of the stomach. So do you guess, okay, what is the infection of the stomach? So just leave, okay, all the components like diarrhea or something, because the infection due to E. coli or Shigella, Salmonella, so all the type of microorganism will cause infection. So this I am talking about stomach only, okay, we are not going to the small intestine and all, okay, in that part, okay, we can discuss. First only the stomach one. So we are going to discuss today an infection caused by a microorganism, okay, a bacteria that is called as H. pylori, Helicobacter pylori. Helicobacter pylori is a bacterium, okay, it's a spiral bacterium, so that may withstand the acidity of the stomach. And usually this H. pylori is not a normal flora of the stomach, and if you are not having the hygienic food intake system, for example, if you are taking the food from outside very commonly, from the hotels or otherwise, and some of the unhygienic conditions, and taking the water from contaminated food based contamination and water, so all these are possibility, okay, to get H. pylori infection. If you are taking the food outside, automatically you will get a lot of complications. Okay, I am not saying, okay, all the restaurants or something. If the restaurant or the food producing factories, okay, if they are not following the stringent or strict the condition of producing the food, the government of India gave a clear instruction to all the food factories and also the food industries like restaurants. They have to follow FFSA norms. Maybe you come across, okay, that FFSA in all the labels and all, okay, they will verify the area of the food production and all. So this H. pylori infection is very common in India, especially in South India, because we are all very prone to H. pylori infection, and so we have normally, okay, we have over acid secretion, because as South Indians we are taking, okay, a lot of spicy food, maybe masala, garam masala, okay, all the spicy food. So this spicy foods will induce acid secretion, and due to that, so all the complication will come, and there is no correlation with acid secretion and H. pylori infection. Once if H. pylori infected to your stomach, automatically that will induce, okay, lot of abnormality in the stomach morphology, skin, that is stomach cell morphology. That means it will produce, okay, and it will attach to the surface of the stomach and produces a lot of hydrolytic enzymes, and it can withstand the acid also. So your acid cannot kill H. pylori. At the same time, okay, this will do a lot of ulceration to the stomach wall, stomach cells, especially, okay, this stomach linings are, and if it is damaged, automatically that may enter into the stomach lining and causes, okay, more infection and inflammation. So this is more painful, okay, if you are getting this H. pylori infection. And initial stage, okay, the acute stage, okay, you will not get any symptoms, maybe some digestive problems, so will may happen, okay, there is no further symptom, okay, in the initial acute stage. Only in the chronic stage you will get a lot of hyperacidity and pain in the stomach. You can feel the pain, okay, in a particular point. Okay, that point is the main point, okay, in the stomach, which is caused by, or ulceration caused by H. pylori. So now we are going to the particular one, okay, that is how we can identify H. pylori infection by using clinical laboratory methods. So this one is going to be dealt, okay, today. So we are going to identify how H. pylori is diagnosed. The infection of H. pylori diagnosis is done, okay, in the lab. Okay, so we can go to the topic. Yeah, can you able to see this window, students? This PDF file. Some of you can unmute your mic because I cannot see the chat box. Unmute
Yes,
your
sir.
yeah. So this window, the
Yes,
clinical
sir.
chemistry. Okay, right. Thank you. And so this one, the diagnosis of gastrointestinal disease is guided by the patient's history and the significant signs and symptoms. So this is mainly by signs and symptoms of the patient, and findings with strong negative predictive values exclude some of the possible causes and focus on differential diagnosis. So the main thing is, so these are all the laboratory investigations used in the investigation of gastrointestinal disorders. So we are going to deal the first one, that is anti-H. pylori antibody titer method, and urea breath test, and endoscopic biopsy with subsequent testing of urease activity and culture histology in the most reliable, okay, investigation. So we can go with H. pylori infection. So what is H. pylori? A spiral shaped bacterium found in the mucous layer of the stomach, okay, in the infected patients. So not very common in all the persons, only with the infected persons, okay, this is going to be present. And H. pylori secretes proteins like some of the proteins may be caused by secreted by H. pylori. This will cause inflammation. Inflammation means this will attract, okay, a lot of white blood cells and causes, okay, swelling and pain, everything. So this is called as inflammation. The inflammation of the mucosa is the first symptom, okay, that means you will get a pain, okay, and redness. If you are seeing, okay, through endoscopy, if you are seeing it, you can identify a redness there, okay. And the enzyme urease is produced by the H. pylori, and this produces ammonia from urea. You can ask the question, sir, how we have ammonia? Because we have the urea, okay, that means in your blood circulation, if it is entering into it, okay, it will get the blood circulation. That means it will get a small secretions from the blood, and it will get only the urea, and urea is converted into ammonia. So ammonia is highly toxic. So this ammonia will cause a lot of ulceration. The main reason behind ulceration due to H. pylori is urease enzyme. So that urease enzyme we are going to use as a tool to identify whether H. pylori is present or not. So coming to the diagnostic test, so we can categorize the diagnostic test into two different types, invasive type and non-invasive type. Invasive means we have to insert some material, like maybe we can open your mouth, or otherwise the doctor can insert a endoscopic tube. So that endoscopy sensor will go inside, and they will take a small sample of the tissue. So that is called as biopsy tissue. So this biopsy can be given to the microbiology lab, and they will culture it. So based on that, they can identify the confirmation. So this need the admission into the hospital, and only in the restricted area with the sterile area they can insert, all of the way they can do this endoscopy procedure and all. And the biopsy handling, you may get a feel of pain, pricking pain at the time of taking a small piece of tissue, okay, from your ulcerated tissue. So these are all the complications, and this is called as invasive. Invasive means to insert something into your body, okay, with the proper help of doctors, okay. And second one is without maybe the doctor, maybe we can do it in the clinical lab. So that is breath test. You can see here, okay, in the breath test, the increase of CO2 14 or CO2 13. So this, what is CO2 13 and what is CO2 14? So this is isotopically labeled carbon. So the CO2, the increase of CO2, okay, the came out of your respiratory system, okay, if you are giving the urea, for example, what is the molecular formula of urea? So this is CO2 CO2 NH2, for example, CO2 CO2 NH2. So this carbon carbon nitrogen. So this is the most excretable form of molecule, okay, in the body. So that is why we are excreting urea. So this carbon dioxide is both the carbon dioxide is coming from TCA cycle and ammonia is coming from deamination of amino acids. So this all the three are most excretable form. Okay, this one is excreted, okay, by the body. Unfortunately, okay, this urea is handleable by the enzyme secreted by H. pylori. So that is a problem. We don't have urease enzyme. If we have urease enzyme, there may be a problem. Okay, and the H. pylori uses an enzyme urease to degrade urea into ammonia. So this ammonia is highly toxic and causes the damage to the cells. Okay, usually the most toxic material in our body is ammonia. Hyperammonemia can kill a person. For example, if you are inhaling ammonia gas, automatically that is a nerve toxin. Immediately stops all the function of a brain, and that is not recoverable also. Okay, that is irreversible damage to the brain. So that is why this ammonia is very toxic, and we are going to use a tool. Okay, that means we are having a chemical substance urea labeled with the carbon either 13 or 14. Okay, so this one is readily available in the market, so we can give the solution to the patient. So let them drink, okay, that urea solution. So don't think, okay, there is a big tube or bottle nearly 1 liter or something. It is only 100 ml solution, okay, given to the patient, and they will drink the urea solution, that is isotopically labeled urea. And after drinking the urea, we have to wash the solution, okay, maybe given in the tumblers or something. Okay, they have to rinse it and again drink it. So because there should not be a loss of urea. Maybe if you are giving the particular dose, means all particular dose, okay, has to reach the body. Clear? So next step is the patient blow the maybe after some time, maybe after the incubation period, usually the 10 minutes of period, you know, there is a meter is there, that is a blower. Okay, this is the blower, and you can give the blower to the patient, and they have to blow the air, that is exhaled air directly from the stomach. They should not pass the breathing air, okay, the air from the stomach. That means only in the depth blow, okay, it may be possible. A patient blows into the sample tube during the urea breath test for gastric ulcers caused by bacteria. So what will happen, okay, this C13 and C14 is liberated as a carbon dioxide, because if you are breaking the urea I told, okay, just now, CO2 NH3. So this NH3 is converted to NH4, okay, there is ammonium ion, and these two CO2 labeled with either 14 or 13 isotopic labeled carbon liberated in the form of carbon dioxide. So the bacteria will liberate urease enzyme. This urease will act on the urea given to the patient, and now the patient will liberate carbon dioxide and ammonia. So that carbon dioxide having C14. So this probe, okay, this blowing tube, okay, having the probe, that probe will detect, okay, isotopically labeled C14 or C13. So based on the intensity of the liberation of carbon dioxide with C14 or C13 or C14, you can identify whether the patient is having H. pylori positive or negative. If it is positive, that will give the breathing, that is signal. If more signal means, that means the patient suffered by a chronic ulcer caused by H. pylori. Okay, based on intensity also we can do it. So this is a very simple test, and unfortunately this is a little expensive when compared to any other test in the world, and so in India, okay, it is not affordable. So we have the test, okay, in the sophisticated laboratories or the high level of the hospitals like Apollo hospitals or otherwise sophisticated laboratories also we have this, but unfortunately not in the common labs. Okay, cost of the testing is 1,500 rupees. So that's all, and maybe, okay, this one is not available all the places, okay, in Indian laboratories. So only in the cities like metropolitan cities like Chennai, Coimbatore, Hyderabad, Bombay, okay, Cochin, Trivandrum, and all the places, okay, we have some of the sophisticated labs or hospitals, they only have this facility. And if you want to do the testing in Pollachi or Thrissur, okay, it is not going to be possible, and you have to go to the higher level of hospitals. So next one is the serum test, the detection of H. pylori IgG antibody in patient serum by ELISA, and third one is saliva test, okay, detection of H. pylori IgG antibody in saliva by ELISA, and fecal antigen test, okay, these are the tests commonly uses that is antigen antibody interaction. So here previously the concept is different, and here the concept is different. So here this concept is related to immunology. You know that, okay, automatically if any pathogen is coming to our body having the immune system, so due to the immune system, okay, we will generate IgG antibody. So have you studied about, okay, this antibodies, different GAMD. GAMD means GAMDE. G means IgG, A means IgA, M means IgM, and D, IgD, okay, GAMD, okay, that means D, and also E, immunoglobulin E. So all the 5 different types, GAMD, okay, this all the 5 different types of immunoglobulins are produced by our body, and a very common one is IgG. So okay, we can use the IgG antibody in the patient serum by ELISA test. So how we can do the ELISA test? So ELISA test is a small vial. Maybe you studied, okay, in the basic immunology or immunology and immunotechnology, and you will study, okay, if you studied in the first year, maybe you know that, or otherwise maybe in the UG level also you studied what are the different types of ELISA, and sandwich ELISA and direct ELISA, okay, like question, okay, because here if antibody towards H. pylori is present in the patient, that means H. pylori is present. So based on that we can do the ELISA test. In the ELISA test, the coated well means we are going to identify how much amount of antibody is present. Okay, so that means we are going to analyze how much amount of antibody. That means the already pre-coated wells with antigens are available, ready-made. So you are going to add your serum, the patient serum, okay, into the tube, and this, if this serum, okay, having antibody, that is IgG antibody, this antibody will bind to the tube's antigen. So antigen is already available in the tube, and the serum is having the antibody. So this antibody, so this antigen antibody complex will be formed, and after that you can wash the tube. So once if it is formed, means, okay, this is a tight interaction, so it will not be detached soon, unless you should not add, okay, any components like urea or otherwise other strong acid or alkali. So in this case, okay, you have to wash with buffer, and after that you can add antibody. Antibody means you already studied, okay, this one, antibody with enzyme or otherwise fluorescent tag, okay, can be present. If you are having, okay, antibody with enzyme, means that antibody will bind to the antibody attached with the antigen, and if it is an enzyme conjugated, means you can pour the substrate to the enzyme, and the colorless substrate will be converted into color. The color intensity directly proportional to the concentration of antigen. If more antigen antibody interactions are there, means more color will be formed. Less antigen antibody interaction is there, means less color. So there is no antibody, means there is no color. So this is very simple concept and very sensitive test also. Okay, this is ELISA test, and the cost of the test is, okay, 1,200 rupees in India. Okay, that is quantitative ELISA I am telling about. Qualitative ELISA only 500 rupees. That is you cannot quantify. For example, the difference between qualitative ELISA and quantitative ELISA means, qualitative means you can give the report that is positive or negative. Quantitative ELISA means based on the intensity of the color, you can quantify how much amount of antibodies are present. So based on that, what is the microbial load? So that is the indirect calculation. So that calculation done by the computer, and finally you have to give the report. That is quantitative ELISA. So based on WHO norms, we have 2 different components. One is qualitative. Qualitative is not a confirmatory test. Quantitative is a confirmatory test as per WHO norms. So next, coming to the saliva test, detection of H. pylori IgG antibody in saliva by ELISA. Maybe in so no need to get blood sample, just we can use the saliva, including, because saliva is a secretion, including saliva having IgG antibody. We can use the saliva, okay, not no need to get the blood from the patient. So we can get the saliva sample, okay, instead of serum, okay, we can use the saliva, that's all. fecal antigen test. The last one is the fecal antigen test. That means the detection of H. pylori antigen in the feces. So this is detection of antigen for the two tests, the serum test of ELISA and the saliva test of ELISA. Okay, that is antibody. We are going to analyze the antibody. So here the last one is analysis of antigen. That means you have the ELISA well and already pre-coated with antibody will be there. So that is ready-made. And what you have to do, get the fecal sample from the patient. So give the small tube and give it to them, let them get the feces, and the feces can be processed, okay, in the lab, and the filtrate of the feces, okay, can be further purified, and only the clear solution can be added, okay, to the ELISA tube. So I am telling again, the ELISA tube, it is not with antigen. In the previous two tests, okay, antigen is coated. Here in the fecal antigen test, antibody is coated. So this antibody coated with coated in the well, okay, the ELISA well, and the sample is loaded now. If you are loading the sample, okay, from the fecal sample, the filtrate, automatically if it is present, maybe if the H. pylori antigen is present, if H. pylori the particles are present, the bacteria is present, so that will go and bind to the antibody. And now you have to use again antibody towards antigen and anti-antibody, and you have to use the substrate for the particular enzyme. So this is also very simple. If more color is means more antigen is present, that means more H. pylori is present in the fecal sample. If less color means less antigen, that means less H. pylori is present. If no color means it is a negative test. So here also we have qualitative ELISA and quantitative ELISA. Qualitative, it will give only the results of maybe positive or negative, and the quantitative ELISA, what is the level of infection. Okay, so that's all about all the different tests available in the clinical laboratory, in the clinical biochemistry laboratory for the diagnosis of H. pylori infection. So we can